I'm going to say welcome to everybody. Welcome to you all joining the March DEBRA UK webinar, Research and Health webinar. We're welcoming Professor Albena Dinkova-Kostova to speak with us today. I'll just quickly run through our housekeeping. This is a recorded webinar. We ask you not to make your own recordings or screenshots. The webinar will be available on our website, and chat or Q&A are enabled. If one isn't working, please use the other one to submit any questions as we go along, and we'll have some questions at the end. There will be a short feedback questionnaire come round. If you can leave just any comments, that'd be great. While I'm doing this, I will advertise next month, which will be Dr. Laura De Rosa, speaking on gene editing in dominant EB, which is EB simplex usually. And I will go back now to welcome Professor Albena Dinkova-Kostova to talk about the therapeutic potential of NRF2 or NERF2. Do we say NERF2? We may be NRF2 activators for EB simplex. So Albena is a professor of chemical biology at the University of Dundee School of Medicine here in the UK, and her research is on understanding inflammation, I think, and the development of strategies for protection against chronic conditions like EB. So you're very welcome here, Albena. I'm going to stop sharing my screen and hand over to you. While you're loading up your screen, I'll do a quick apology. I was always told not to start with an apology. At the time, standing in for Sagir Hussain, who can't be here today. So welcome, Professor Dinkova-Kostova. Please begin your webinar.
Okay. I hope you can see the screen and the presentation.
Brilliant.
Great.
Just to let you know, yes, it's all good. And now I'm going quiet.
So thank you very much for the invitation and this opportunity to tell us, to tell you how we got into this field. And I will begin from my expertise, which is on this transcription factor, NRF2. It is a protein, a transcription factor, which means that it regulates the expression of genes that have specific regulatory sequences on their DNA. And NRF2 has been shown to be protective in multiple animal models of human chronic diseases. And as you can see here on this slide, these involve diseases of the central nervous system, such as Alzheimer's, Parkinson's disease, ALS, also diseases of the eye, liver, heart, kidney, as well as lungs. And I have added to this picture the skin, and I will show you some examples of the work that we've done in this context. So as I mentioned, NRF2 is a transcription factor and a protein which usually is degraded very quickly when it's produced. And here I have shown it as this purple molecule. It binds very quickly to this dimeric protein, the blue protein, which is called KEAP1. And KEAP1 has a function to mediate a specific post-translational modification of NRF2 called ubiquitination. So it mediates the addition of these chains of the small protein called ubiquitin. And when that happens, this is viewed and recognized by the cell as a sign for degradation, which is done in a particular organelle called the proteasome. So in a normal cell, under perfect conditions, NRF2 is made and very quickly degraded. So it only lasts in the cell for about 10 to 20 minutes. And you would ask, and so did we initially when this pathway was discovered, why would the cell invest so much energy in making a protein only to then destroy it so quickly? And this is because this mechanism allows the cell to very quickly, when there is a sign of danger, for example, to inactivate this process, making NRF2 immediately available. And this can happen Also, if we are exposed to certain chemicals from our environment or from our diet, these chemicals can inactivate KEAP1, which means that this complex becomes dysfunctional. Consequently, NRF2 cannot be degraded, and the protein that is synthesized then becomes stabilized. It translocates to the nucleus and activates the transcription of many genes. We think that there are on average of 500 different genes that are regulated in this way. And by and large, these genes encode proteins with protective function. So we call them cytoprotective genes. So this function of NRF2 allows this initial danger signal to be recognized and eliminated, essentially, before it has a chance to damage the cell. We can monitor this process of KEAP1-mediated NRF2 degradation, for example, in live cells by this, what is known as an imaging experiment, where these cells now are engineered to express KEAP1, which is fused to a green fluorescent protein, which has green color when we look at it under the microscope. And then they also express NRF2, which is labeled in red. So this is the ideal homeostatic state of the cell. We only see green because KEAP1 degrades NRF2 by the mechanism that I described. However, when the cell is exposed to a certain compound, which inhibits the dysfunction of KEAP1, then with time, we see the increased accumulation of the red signal, which indicates that NRF2 accumulates. And I've just given one example to illustrate the protective nature of this response. In this situation, the cells have been exposed to different concentrations of a damaging agent. And then the percentage of the cells that are still alive has been measured. And you can see in these white bars that at this dose of the damaging agent, only 20% of the cells are living. However, if the cells were, same cells were pre-treated with a chemical which activates NRF2, you can see that now most of the cells are alive. And this really illustrates how potent this response and how powerful it is and how important it is for cell protection. Now, very interestingly, we have some of these chemicals that can activate NRF2 in our diet. And one example is this molecule called sulforaphane. Sulforaphane is present in a family of vegetables known as cruciferous vegetables. And you can see here broccoli, which are quite a rich source of sulforaphane. But what is interesting is that the plant produces a precursor of sulforaphane called glucoraphanin. So this molecule is completely biologically inert. It has no reactivity. But the plant also makes an enzyme called myrosinase in a different cell of the same plant. And it's only when the plant is injured, for example, when we chew on the broccoli, when this enzyme can get in contact with its substrate. And very quickly, this enzyme promotes the formation of the product, sulforaphane, which is very biologically reactive. And this picture shows a plate with broccoli sprouts in it. So these are 3-day-old sprouts from broccoli. So the seeds are grown only for 3 days. They give rise to this very young plants. And what is interesting is that these sprouts have 50 times on average higher content of this glucoraphanin compared to the mature plant. So it seems like plants produce these phytochemicals, as we call them, very early in their development, and then their content gets diluted as the plant matures and grows. Now, we've seen that sulforaphane can activate NRF2 in cells, and this is an example of skin cells isolated from mice that were exposed to sulforaphane. And with the different increasing concentrations of sulforaphane, you can see this NRF2 activation process. This process is measured by the gene expression of one of its target genes called quinone reductase. This does not happen if we have cells from mice that are null for NRF2, suggesting that this activation requires NRF2. We can see the same if we topically treat the skin of live mice, and then after 24 hours, we can sacrifice the mice and then make skin homogenates and measure again NRF2 activation by its target gene NQ1. And again, you see this increase in its target gene in wild type mice, but not in NRF2 knockout mice, suggesting that at least with this type of activation, it is due to NRF2 and not any other function of sulforaphane. Now, what about, is it protective? Again, in this experiment, which used cells from mice, wild-type mice, these were skin fibroblasts that were exposed to ultraviolet radiation as a relevant damaging agent to the skin. And one consequence of exposure to ultraviolet radiation is the generation of reactive oxygen species, which can cause oxidative stress. So we can measure those in the lab, and you can see here in the control cells, with increasing doses of ultraviolet radiation, there is increased levels of reactive oxygen species, which could lead to oxidative stress. However, if we pre-treat cells with sulforaphane, this is greatly suppressed. And that also improves cell viability in a similar way to the example that I showed you earlier. Now, I told you that sulforaphane activates NRF2, and we can also measure downstream of this oxidative stress another potentially damaging process, which is inflammation. And if we again expose cells to sulforaphane and also challenge them with an inflammatory agent, you can see that as NRF2 activation proceeds and increases, the inflammatory response decreases. And because inflammation and oxidative stress are involved in part in the photodamage and photocarcinogenesis by UV light, here we had done an experiment in mice where we exposed them to ultraviolet radiation as a relevant human carcinogen to the skin, leading to skin cancer. And the exposure is quite relevant to human exposures. It doesn't really cause any sunburn, but we expose them chronically for about 10 weeks. And by the end of this exposure to ultraviolet radiation, they are still no mice with tumors, because tumorigenesis takes much longer to develop. But in the subsequent 10 weeks, you can see that most of the control mice, so these are mice that were exposed twice a week intermittently to ultraviolet radiation, they do develop tumors. And almost all of them, if we extend this period, 100% of the animals will develop at least one tumor. However, if we start treating with, in this case, we use broccoli sprout extract as a source of sulforaphane after UV exposure, then you can see that we can protect to quite a significant degree the incidence, which is the percentage of mice with tumors in each group. Also, we can decrease the number of these tumors, and the biggest effect is actually on the tumor volume. So even those mice that develop tumors have much, much smaller tumors if they had been treated with sulforaphane. Now, NRF2 activation itself is protective. And in this experiment, we have used mice that have a low or normal level of NRF2, but also transgenic mice which have high levels of NRF2. So again, these are engineered to have at all times high levels of NRF2 without the need for chemicals such as sulforaphane. And you can see that similar to the protection of sulforaphane, again, these mice with high NRF2 levels are protected. In fact, we let them live much longer because they were simply not developing or hardly developing any tumors. This is one tumor per mouse at most. This, I'd like to just stress out that in this model, the mice develop cutaneous squamous cell carcinoma. So these are skin tumors that are the most common tumors in people. And this is a section that was examined by a human pathologist of tumors from our mice. And it was really nice to hear that he told us that these are exactly the tumors that he sees in his patients in the clinic. So they have these very characteristic keratin swirls in the middle, and this is the invasive edge of the tumor. We have subsequently also performed whole exome sequencing and other DNA methylation analysis to make sure that this model is really recapitulating the human disease. Now, these are all mice. What about humans? So the advantage, of course, of sulforaphane being found in broccoli is that we can make a broccoli, and we use broccoli sprouts, in this case, extract, and apply it topically to the skin of humans. Since sulforaphane is present in the diet, ethical approval to apply topically to the skin was quite straightforward to obtain. And we did a very similar experiment now, applying it topically to very tiny areas of skin, about 1 centimeter in diameter, then taking little 2-3 millimeter punch biopsies and measuring again the cytoprotective or defense genes that NRF2 regulates. In this case, NQO1 and this other one, AKR1C1. And you can see that in both cases, when we apply this sulforaphane containing extract, we can induce this cytoprotective response, not just in mice, but also on the human skin. Is it protective? So to answer that question, we decided to use UV lamps that are used for phototherapy in dermatology, and had to design some sort of template allowing us to apply very small amounts of sulforaphane containing extracts. These are 1 centimeter circles. We had to design this template. This is an adhesive material that we put on the back of a subject. These are all healthy volunteers. Then we can apply to some of these windows the sulforaphane extract, and to the others we used the vehicle control. We did this application in this zigzag manner because, you can see here the lamps in the unit ran parallel to the body. So we didn't want to have any bias of perhaps any weaker lamp for comparison. So that's why they were in this zigzag manner. Then the subject can be completely covered, and we can have shutters on these windows so that we can expose only one at a time, or as many as we needed to the UV radiation. And when we did that, you can see this is a picture of one of the subject's backs. after the application of different doses of sulforaphane, and visibly you can see that the sunburn, it's not a sun, it's a UV light, but this redness was decreased in almost all cases where sulforaphane was applied compared to the solvent. We can quantify this redness using a handheld device called chromometer, which is used to measure redness. And then we can plot here the percent reduction in skin redness, and you can see that sulforaphane application can protect, not fully, but at about 60% we get saturation in the protective response. Now that was a proof of principle studies. Subsequently, we have done a proper randomized double-blind placebo-controlled study in healthy human subjects again, where this is the study design here. Again, we apply these broccoli sprout extracts to the back of healthy subjects, and we use half of the bag to apply sulforaphane and the other half glucoraphanin. So this is the precursor of sulforaphane, the one that has no biological activity, but at least we were able to match the color because we didn't want both. As you know, plant extracts have some color, it's a yellowish green color, and we didn't want to bias ourselves and potentially filtering the UV radiation due to this pigment. So in this scenario, again, with the placebo-controlled trial, we saw less redness in the sulforaphane group compared to glucoraphanin. Now, you probably wonder what does it have to do with EB? And this is when we notice that in addition to these defense genes that I showed you are regulated by NRF2, actually NRF2 activation and sulforaphane also leads to induction of some keratins. And here is an example of keratin 17 and keratin 16, the levels of which are increased after sulforaphane exposure, and these are keratinocytes. Not all of them are increased. Some of them, for example, K14 doesn't change, but K17 definitely does. This is the messenger RNA level, and this is at the protein level. Now this brings me to EB. And as you know, EBS specifically is a group of autosomal inherited diseases. Fortunately, the incidence is quite rare, and it's characterized by these fluid-filled blisters that appear after mechanical trauma that heal without scarring. And this process, of course, is very painful, very debilitating. Many of the patients, I am told, end up on wheelchairs eventually because it is impossible really to walk. And this is an electron microgram of a blister from this particular patient. Again, as you know, EBS is a subset of EB. These are monogenic mechanobullous dermatosis that are characterized by extensive skin fragility. And just again, for this audience, it's not necessary, but to remind you that EBS, the location of the blisters, is in this basal layer of the keratinocytes when we consider the skin architecture as a whole. And at the molecular level then, the disease is caused by dominant changes in the sequence, as we call them mutations, in either keratin 5 or keratin 14 genes. And these two keratins copolymerize to form cytoskeletal intermediate filaments. And by doing that, they really provide a very strong network that is responsible for having the skin being so strong normally. And when there are alterations in either of these, then the skin becomes very fragile. Now, fortunately, there is a functional redundancy in many of the keratins, And in this paper, for example, they made a mouse that completely lacks keratin 10, which is one main keratin. And somewhat surprisingly, they saw that actually the skin looks quite normal. So this is the skin of the K10 null mice in comparison to wild type. But the difference is that these keratins, 5, 14, and 15, which normally are only expressed in this basal, the very bottom layer of the keratinocytes in the skin, now they are expressed throughout the skin of keratin 10 knockout mice. And this suggests that by increased expression of all of these keratins, they can compensate for the lack of keratin 10 and still restore the normal skin architecture. So I just bring you back to the same slide that I showed you a while ago. And in this context, we then asked, could sulforaphane-mediated induction of keratin 16 and 17 that we observed compensate for the absence of keratin 14 in a mouse model of EBS? And this was done in Pierre Coulombe's lab, who was one of the discoverers of the genetic causes for EBS when he was a PhD student with Elaine Fuchs. And in this experiment, we treated pregnant K14 null mothers in the last week of pregnancy. So these were 3 treatments with sulforaphane just before the pups were delivered. And then we measured the expression of keratin 17 in the skin of the newborn pups. And again, just similar to the cell treatment that I showed you earlier, we saw an increased expression of keratin 17. This is the forepaw and this is the tongue. And then we also looked for blisters, and these are the blisters that are formed in the newborn mice. They are very quickly developing these blisters. Just the act of being born promotes this blistering. And they were completely protected, those whose mothers were treated with sulforaphane. And then Michelle Kearns, who was a technician in Pierre's lab at the time, was wonderful in terms of continuing to immediately topically treat the skin of these newborn pups. And these are the pictures. So these are the control wild type mice. And these are now the sulforaphane treated. And you can see that in the control, this is just a paw enlarged picture of this one, of 2.5-day-old pup, this severe blistering phenotype. This is how a normal pup should look like. And then with the sulforaphane treatment, this was almost completely eliminated, the blistering phenotype. And actually at this day, at 4.5-day-old, we could only compare the sulforaphane treatment with the wild type mouse, because most of these pups with EBS disease were already dying, mainly because of the oral blistering that they develop. They are unable to feed, and also their thermoregulation is disrupted. So for various reasons, we moved away from sulforaphane. And I just want to say this is not because most people don't like broccoli. And one of the reasons actually was that during that time, we started a collaboration with 2 teams of these are the biologists, Karen Leiby and Mike Sporn, and chemists Tadashi Honda and Gordon Gribble, who were at Dartmouth College in New Hampshire, and they were developing compounds of this sort. So this is another derivative of a natural product called oleanolic acid, where the chemists have put functional groups on these rings of these molecules. And together with them, we've discovered that, first of all, they are very potent NRF2 activators. Some of them are much more potent than sulforaphane, hundreds of times. And also, similar to sulforaphane, there is a very good correlation between the ability to activate NRF2 and suppress inflammation. Now, 20 years later, it's quite gratifying to see that this particular derivative, called omaveloxolone, is now used clinically, and it was approved in 23 by the FDA and subsequently in 24 by the European Medicinal Agency as well for the treatment of Friedreich ataxia. As you may know, this is a devastating, another rare disease. There is no treatment for it, and this was really a big breakthrough that there is something now for these patients. The disease is caused by gene alterations in a protein called frataxin. It's located in the mitochondria, and these alterations lead to mitochondrial dysfunction, which then leads to reactive oxygen species, oxidative stress, inflammation, and in a very similar vein to what I've been discussing, because of this anti-inflammatory and antioxidant effect of NRF2 activation, this compound now has benefits in these patients. So this compound was actually, during its development, was also developed as a topical agent for skin application. And in this paper, they have treated rat skin with this molecule applied in different cream preparations and monitored NRF2 activation. And the darker stain suggests higher NRF2 activity in the epidermis, and these are the hair follicles of the rat skin. As part of this study, they also applied it to human skin, and they found that there is certain level of NRF2 activation. Again, the darker the stain, the more activity. They tried different dosing regimens. These were for 2 weeks daily doses on different areas of the skin. And then this was a longer term treatment, almost 1 month, 28 days, the purpose of which was to see whether there would be any potentially side or unwanted effects, which they didn't see. So that was really nice and encouraging that the compound seems to be suitable for topical application to the human skin chronically with daily dosing. Now, obviously, we can't use human skin in living subjects very readily, and specifically for mechanistic understanding, we need another system. And this system is called TenSkin device, where it was developed by Robin Hickerson in Dundee, who worked with Irwin McLean at the time. And they now have a spin-out company called TenBio, where they take discarded human skin, usually from tummy tucks or breast reduction surgeries, and they place it in this device, which then provides a way for the human skin to be manipulated in an ex vivo setting, so outside of the body, but under a proper tension. Because if you take a piece of skin just as an explant, obviously, and you can keep it viable for a few days, but it forms a ball in a petri dish, essentially. So it's not physiological, and the responses won't be completely normal. So in this way, they have shown that they can keep the skin in a proper environment. In this device, this is media from which the skin cells are fed. You can apply topically, as exemplified here, compounds to the surface. You can also sort of mimic systemic exposure if you apply a compound of interest in this tissue culture media. And then here is a picture of skin that has been maintained in this condition for 2 weeks, but it is also viable much longer periods of time. So using this system, Robin Hickerson applied this compound, omaveloxolone, and she then looked at keratin 17 expression. And you can see again that it seems that omaveloxolone does increase the expression of keratin 17 as shown by the red color. And these blue dots are the cell nuclei, and this is the staining around them. Now, Aileen Sandilands, who is a postdoc now in my lab, has also looked at the ability of omaveloxolone to induce the expression of keratin 16, now in a EBS cell line. The previous experiment was done on normal, healthy skin. This is now EBS cell line that bears this is one of the severe version of EBS, this arginine 125 to proline change in keratin 14. And this is just a classical NRF2 target for comparison. So as we increase the concentration of omaveloxolone, we see more and more NRF2 activation as measured by this downstream classical target and QO1, but we also are beginning to see keratin 16 increase in keratin 16 expression. So we are very optimistic that this suggests that hopefully even in the context of mutant K14, which is highly relevant to the human disease, we could hopefully achieve compensation by keratin 16, possibly 17. So we just started this project, and we are most grateful to DEBRA for funding it. And I just wanted to share with you at the end that our main research question is whether we could repurpose omaveloxolone for patients with EBS. Our hypothesis is that by inducing the expression of keratin 6, 16, and or 17, omaveloxolone will compensate for the mutant keratin 14, correcting the defective cytoskeleton, and thereby reducing skin fragility, and also by decreasing the levels of reactive oxygen species and inflammatory mediators, which this compound and NRF2 also do, we could hopefully achieve alleviation of inflammation, which should further contribute to reduction in skin fragility. So our aim is to determine whether omaveloxolone induces these keratins and reduces inflammation. First of all, in normal human skin, using this TenSkin tension-based human ex vivo skin model, and also in collaboration with our partners, co-applicants in the Netherlands, co-investigators who have access to EBS patient skin, and are developing a 3-dimensional organotypic human skin equivalent model to see whether we can achieve this effect in the context of the disease. So this is our collaborator in Groningen, Peter van der Akker. This is the postdoc in Dundee, Aileen Sandilands, and this is our collaborator, Robin Hickerson at 10Bio, who will be providing those TenSkin devices. I'm very grateful to all of them for their effort so far on the project, and I look forward to continue collaborating with them. We also are most grateful to DEBRA, as well as their Dutch equivalent, the Dutch Butterfly Child Foundation, who are jointly funding this project. I also want to acknowledge the contributions of many people who I've had the pleasure to work with over the years, who have contributed in many ways to this project. Paul Taole was my mentor at Johns Hopkins, and we really started looking at human skin and NRF2 with him. Pierre Coulombe, who did the study in the keratin 14 knockout mice, together with Michelle Kearns. Michelle was a young 21-year-old technician who treated all of these animals and really rescued them. Now she went to medical school subsequently and is a dermatologist at Johns Hopkins University. These are two of the postdocs from my lab who have contributed to the skin carcinogenesis studies that I mentioned. And I also would like to thank Erwin McLean. You are probably very familiar with him. He worked with DEBRA funding for many years, really excellent skin geneticist. And Birgit Lane, again, she was in Dundee and she discovered the keratin mutations in EBS. And thank you very much for your attention. I'll be happy to take any questions.
Thank you so much for that, Albena. I really enjoyed the clarity of your presentation. So thank you. Quite a lot of science in there, but very clearly laid out. I like it. So if anyone has any questions, please do type them into the chat. I think I've enabled it so everyone can do it, but I would appreciate it if somebody just typed yes, so I know that at least you can ask a question if you want to. Oh, thank you, Karen. That's fantastic. So I did have a couple of questions, but they were sort of building as I went along. With the mice. So there is an NRF2 knockout mouse.
Yeah. Yeah.
Does it have a normal lifespan? Does it have any symptoms?
Yes. So that's a very good question. It looks normal. The only phenotype when you look at the mouse is that mice have quite dark stained teeth. The NRF2 knockout mouse has white teeth. So that's the only obvious phenotype. And this is because NRF2 is also involved in iron homeostasis. Now, it looks normal. However, in all the disease models that this animal has been into, the disease pathogenesis in every disease is hugely accelerated. For example, if a model in normal mouse takes 2 months to develop the disease for, let's say it's a liver disease of some sort, caused by high-fat diet, for example, we can see steatotic liver at 2 months. In NRF2 knockout, we see that at 2 weeks. So there is a very profound acceleration of any disease. And we think it's because this initial response that allows adaptation and suppression or elimination even of the initial insult, disease-causing insult is not there to protect. So that's the main.
They're normal unless something bothers them, and then they're much more vulnerable. That's very interesting. And I should probably know, has anybody crossed them with any of the EB mouse?
No.
I think we can imagine what the outcome might be, but it's I know that for people living with EB, we really don't want to go down a rabbit hole of trying all sorts of different preclinical things. As soon as we get to this point where we think this could be a useful treatment, we do want to advance it as quickly as possible. And it does seem that that's what your project is doing. Do you have any idea how long it might take to get some results that would lead into a clinical trial? It's an approved. Omaveloxolone is approved, so
yes, it's an approved drug now, recently approved from 2 years ago. So there are a couple of unknowns. I think in terms of how long it will take, our clinical colleagues are ready to start a trial. They tell us right away. However, we do not know. First of all, the drug is approved for oral indication. So that we don't know whether by oral route there will be sufficient induction of the keratins in the skin. So that's completely unknown. So I think we need to have some insight. So we've been discussing perhaps the idea of requesting, if it were possible, some of the Friedreich ataxia patients who have been treated for a long time to take some skin biopsies from them to see whether that's a feasible route. Topical application, I think, is also potential, but that will require development of a proper delivery vehicle. I mean, this paper that I showed some results from where they used healthy human skin and developed a cream. If we can get some insight, that study was done by a pharmaceutical company, which doesn't exist anymore because when they got the approval, they sold
It often
immediately.
happens. The names change all the
So
time.
if we could get some insight what sort of cream that was, I think that will be immensely helpful. For the moment, we are planning to, in this 10 skin device model, to compare application topical to the skin as well as in the media. But going to patients, because their skin is so much thicker due to this constant blistering and healing, it might be even more challenging to develop a topical treatment. So there are all of these little questions that are so important to understand how long it could actually take to know the answer. But we do collaborate, especially Peter in Groningen. He's a clinical geneticist, but we have a collaborator on the project who is a dermatologist, and she deals with these patients on a daily basis. So yeah, we are very
Brilliant.
much.
That was a very measured answer. I think, although people want to hear someone say, yes, it'll be ready in how many years, I think it's very honest to say there are challenges always with getting these, even the substances that we have high hopes for, to get them to the right place in the skin. We recently heard from some, I don't want to betray any confidences, but different methodologies. We've heard about nanoparticles and microneedling and
Yeah.
nanoneedles. Would it be fair to say that the delivery is a slightly separate branch of science to the
Yeah. Yeah,
yeah.
actually, that was one of the reasons why we didn't progress with sulforaphane, because basically we were at that stage where we needed a good way of delivering it to the skin in all of these contexts that I described, all of the sort of even the clinical trial, because it was done as phase 1 on healthy subjects. We used acetone, which you cannot use in any disease context. So to try to develop a good delivery vehicle, basically we were stuck there because sulforaphane is a natural product, so there was no possibility for patenting or anything like that. That was a big impediment as well.
Yeah, I mean, my understanding is our skin exists to keep everything out, so
Yeah.
trying to get anything in, even therapeutically, is difficult. There is a comment, it's not a question, but just from one of our attendees who congratulates you and would like to get in touch about this, James Dixon.
Okay, wonderful.
Yeah,
Thank you.
yeah. So if we're doing nothing else, we're bringing our researchers together. I think unless there are any questions, burning questions that people would like to type in very quickly, I'm going to wind us up and give people 10 minutes back of their hour. Thank you so much for really excellent presentation. I certainly enjoyed it, and it gives us some hope, I think, that there are so many different approaches to tackling the symptoms in EB. I will say thank you to everyone for attending. Thank you so much. If you have any extra questions, there'll be a feedback form will be circulated by Zoom. Please do just add them in where you get the option to leave a comment. Thank you so much, Albena. It was really great to hear from you. And thank you to everyone for attending. I hope to see you all, or sort of be aware of your existence on the call next month. Thank you very much again, and
Thank
take care,
you.
everybody. All the
Thank you.
best.